This lesson's on proteins, and here are your objectives. Now first, it's important to recognize just how important proteins are. They actually make up most of the dry mass of a cell, and they perform virtually all of the functions that you can think of in a cell. They can be catalysts, like the enzymes that we'll learn about in a future lesson. They can be structural. They can be involved in transport, like the pump proteins or the diffusion carriers that we have learned about already. They can be actually motors carrying things around in a cell. They can be involved in signaling. They're involved in immune function or defense. They just have a myriad of different functions, all of which depend actually on their three-dimensional shape. So we'll start to learn about how they get that three-dimensional shape in this lesson. Let's clarify two different terms. A polypeptide is a polymer of amino acids. So as we will see, proteins are made by stringing amino acids together using bonds called peptide bonds. And if you just string a bunch of amino acids together using covalent bonds, then you get a polypeptide. Proteins can be made of one or more polypeptide. So you can have one polypeptide proteins or multiple polypeptides in a protein, and they assume a 3D shape, or what we call a conformation. And that 3D shape, again, is going to be what's important for the protein's function. The sequence of amino acids in a polypeptide, and then as we'll see, therefore, the shape of the protein, is determined by the DNA sequence. Or, in other words, it's determined by genes. And we'll learn about how that process happens in a future lesson. Let's look at the structure of one individual amino acid. An amino acid has a central carbon that gets referred to as the alpha carbon, and then around that, all amino acids have one hydrogen group, one carboxyl group, like this, one amine group, which looks like this, and then, so all that part is the same, and then the part that varies is called the side chain or the R group, like that. And this is the part that varies. Otherwise, everything that I've drawn in blue there stays the same. And there are 20 different amino acids that make up proteins in general. And these 20 amino acids are joined together by covalent peptide bonds to make a polypeptide. So if I redraw two generic amino acids here, and I get rid of water like this, then I end up with C, O, N, and H, like so. C double bonded to O, single bonded to N, and an H. So this bond here is called the peptide bond. So what we've seen is a dehydration or a condensation reaction between the carboxyl group of one amino acid and the amine group of the other, again releasing water and forming a peptide bond. Notice also that you get this backbone of repeating nitrogen, carbon, carbon, NCC, where the first C is the alpha carbon and the second C is the carbon from the original carboxylic acid group. Sticking out from this repeating backbone, you have the R groups, or the side chains. Let's take a look at some of those side chains. This table is from Campbell Biology, showing the 20 amino acids. Notice that they're grouped into different categories. First, we have the hydrophobic side chains. Those are ones that are going to want to hide away from water, and therefore they'll be found on the interior of a protein, because proteins are an aqueous environment in our cells. We also have hydrophilic side chains that can be either polar or actually electrically charged, either positive or negatively charged. And interactions between the hydrophilic side chains and water will tend to put them on the outside of proteins. Also, the attraction between positive and negative ions in the side chains of a protein can help to stabilize the overall three-dimensional structure. Hydrogen bonds between polar side chains of different amino acids in a protein can also help to hold it in a particular three-dimensional structure. And one last amino acid, a special case to be aware of, is cysteine, which has this SH or sulfhydryl group on it. If two cysteines react to get rid of those hydrogens, then the sulfurs can actually be bonded together. Each of these belongs to a different cysteine in a different part of the protein, and that forms what's called a disulfide bridge, and that's a particularly strong, because it's covalent, bond that can hold the protein in a particular shape. So there's a variety of different interactions that the side chains can have with each other that help to hold proteins in particular 3D structures. That's an introduction to the structure of proteins.